Hello everyone, my name is Gallagher Mui. Today, I'll be presenting my project on the druggability and genetic variability of the influenza A proteome. This project was conducted under the supervision of Dr. Shapira. The influenza A viral particle is consisted of 8 segmented negative-sense single-stranded RNAs. These 8 RNA segments encode for 10 major proteins, including the transmembrane proteins hemagglutinin, neuraminidase, and M2, PB1, PB2, and PA, which together form the RNA polymerase complex, nuclear protein, which binds to the RNA segments, M1, which oligomerizes underneath the lipid bilayer and is attached to the viral RNAs, and finally, ns1 and ns2. The viral ribonuclear protein complex is comprised of the RNA segments, the 3 subunits of the RNA polymerase, and nuclear protein. The replication cycle begins when the hemagglutinin protein of the influenza A virus recognizes and attaches to the sialic acid molecules on the host cell. Endocytosis of the influenza A viral particle then takes place, and the influenza A viral particle is packaged inside vesicles. In the late endosome, the acidic environment activates the M2 ion channels of the influenza A virus. This allows an influx of protons from the endosome into the interior of the viron, resulting in the dissociation of viral ribonuclear proteins from M1. Concurrently, a conformational change in the hemagglutinin protein results in the exposure of a hydrophobic fusion peptide. This fusion peptide is inserted into the endosomal membrane and allows for the fusion between the endosomal and viral membrane. The viral ribonuclear proteins are then released into the cytoplasm and are exported into the nucleus of infected cells. In the nucleus, the heterotrimeric RNA-dependent RNA polymerase complex catalyzes both the replication and transcription of viral genes. Several accessory and non-structural proteins, including NS1, PAX, and PB1F2, are also generated via alternative splicing. After replication or transcription, the newly synthesized viral RNAs and proteins are exported out of the nucleus and into the cell membrane, where new viral particles are assembled. The neuraminidase protein, which cleaves sialic acid molecules of the host cells, facilitates the budding and release of viral progeny. The released viral progeny can now infect a different cell. The influenza A virus is classified based on the composition of 2 surface proteins, hemagglutinin and neuraminidase. There are 12 of 18 subtypes of hemagglutinin, H1 to H18, and 9 types of neuraminidase, N1 to N9. Each unique matchup of the hemagglutinin and neuraminidase subtype results in an antigenically different pathogen. The influenza A virus normally resides in animal reservoirs, such as swine and avian species. However, the influenza A viral genome is known to change continuously in 2 separate processes called antigenic drift and antigenic shift. This results in the unique combination of the hemagglutinin and neuraminidase subtypes, and on occasion, the influenza A virus gains the ability to infect the human population, resulting in large-scale epidemic outbreaks. The influenza A virus have caused some of the most severe pandemics in recent history, including the Spanish flu in 1918, the Asian flu in 1957, the Hong Kong flu in 1968, and most recently, the swine flu in 2009. There are also seasonal flu outbreaks that are prevalent during the winter. According to the Centers of Disease Control and Prevention, during the 2019 to 2020 flu season, over 38 million people were infected with the influenza A virus in the US, and 20,000 deaths have been estimated for that season alone. This shows that the influenza A viral infection is a serious health concern. The best measure in preventing an influenza outbreak is through vaccination. However, as previously described, the viral genome is known to change continuously, and therefore there is a constant need for vaccine reformulations. The second option is through the usage of antiviral drugs. Currently, there are two classes of antivirals that are approved in Canada. The first class of drugs is the M2 channel inhibitors. An example of such is Emerity. However, this drug class is no longer recommended for usage because recent circulating strains are largely resistant against it. The second drug class is the neuraminidase inhibitors, which include oseltamivir, cinnamivir, perimivir, and lenimivir. Due to the heavy reliance on this drug class, there have been concerns that mutations that confer drug resistance will arise. During the 2009 swine flu pandemic, the circulating strain at that time was identified to have a histidine 274 tyrosine mutation in the neuraminidase protein. Influenza A virus with this mutation displayed increased resistance against the inhibitor oseltamivir, but not against other neuraminidase inhibitors. Regardless, mutations against other neuraminidase inhibitors will eventually develop due to our heavy reliance on them. To prepare for the next influenza A pandemic, drug discovery efforts should focus on the development of pan-influenza A inhibitors. The objective of this project is to identify binding sites in the influenza A proteome that are both druggable and conserved across all influenza A strains using in silico methods. With respect to the druggability of binding sites, we assume that not all binding sites are biologically relevant and that not all biologically relevant binding sites can be targeted by compounds. Factors that could influence the druggability of a binding site include the hydrophobicity of the residuals lining the pocket, the size and enclosure of the cavity. With respect to the genetic conservation of binding sites, we assume that residuals lining the binding pocket that are conserved across all influenza A viral strains are under strong selective pressure. Therefore, these residuals are unlikely to accumulate mutations throughout time, and the binding pocket serves as an ideal target for the development of a pan-influenza A inhibitor. Conversely, if residuals lining a binding pocket are highly variable across all influenza A viral strains, mutations in these residuals will likely accumulate, and the binding pocket would not be an ideal target for the development of a pan-influenza A inhibitor. To determine the druggability of a binding site, we first derive protein structures from the protein data bank. We then visualize these structures using ICM-Pro. Potential binding pockets were determined using ICM-Pro's pocket finder function, and the residuals surrounding the binding pockets were identified. Finally, the druggability of the binding pocket was assessed using sitemap if no inhibitor was known to bind to that particular binding site. To determine the conservation of residuals lining the binding pocket, the protein sequences of all influenza A viral strains were first downloaded from the NCBI influenza virus database. A multiple sequence alignment was then performed using KLI, and the amino acid variation, percent identity, and percent conservation of residuals lining the binding pocket across all influenza A viral strains were determined. We also evaluate the change in binding free energy upon mutation in the residuals lining the binding pocket using the tri-mutation function in ICM-Pro. The first protein that will be discussed in detail today is the neuraminidase protein. The neuraminidase protein is a homotetrameric protein that is found on the surface of the influenza A viral particle. Each monomer is consisted of a cytoplasmic tail, transmembrane region, stalk, and Gallagher head domain. Here, the structure of the influenza A head domain is presented. Using ICM-Pro's pocket finder function, we identified 5 potential binding pockets in the neuraminidase protein. 2 of these pockets are biologically relevant. The Gallagher pocket, as previously discussed, possesses sialadase activity, and it facilitates the budding and release of viral progeny. The Gallagher pocket is present in all neuraminidase subtypes. Here, the 18 residuals lining the Gallagher pocket are displayed. The 150 cavity, on the other hand, possesses no catalytic activity However, due to its close proximity to the Gallagher pocket, the 150 cavity may allosterically regulate the former's activity for ligand binding. Unlike the Gallagher pocket, the 150 cavity is not present in all neuraminidase subtypes. The cavity is present in group 1 neuraminidase, including N1, N4, N5, and N8, but is absent in group 2 neuraminidase, including N2, N3, N6, N7, and N9. Here, the 15 residuals surrounding the 150 cavity are shown. We next examine the druggability of the 150 cavity. Using sitemap, we determined a druggability score of 1.007 for the 150 cavity. This indicates that the 150 cavity can be in fact targeted by ligand binding. We did not assess the druggability of the Gallagher pocket using sitemap, as this pocket is universally recognized to be druggable. Several neuraminidase inhibitors are known to bind to this Gallagher pocket. Here, the structure of neuraminidase in complex with cenobamate and oseltamivir are shown. For the genetic conservation analysis, we determined the percent identity conservation and the sequence variation of the 50 amino acids lining the 150 cavity of the neuraminidase protein. 3,469 influenza A viral strains were used in the multiple sequence alignment. Percent identity and or percent conservation with a value of over 99% are highlighted in red. 7 out of 50 amino acids lining the 150 cavity show a percent identity of over 99%. And 9 out of 50 amino acids show a percent conservation of over 99%. Here, we determine the percent identity conservation and the sequence variation of 18 amino acids lining the Gallagher pocket. Similarly, 3,469 influenza A viral strains were used in the multiple sequence alignment, and percent identity and or percent conservation with a value of over 99% are highlighted in red. 16 out of 18 amino acids lining the Gallagher pocket of the neuraminidase protein show a percent identity of over 99%. And 17 out of 18 amino acids lining the Gallagher pocket show a percent conservation of over 99%. From the previous figure, we observed that the residue 246 lining the Gallagher pocket of neuraminidase display the lowest percent identity and conservation among other residues lining the Gallagher pocket. In a separate study, it was shown that the neuraminidase subtypes N1 and N4 contain serine as its position, whereas N5 and N8 subtypes and all group 2 neuraminidase contain alanine at this position. We would like to determine whether this subtype specific genetic variation may affect ligand binding or not. The residue of our interest, 246, forms a hydrophobic pocket along with isoleucine 222, arginine 224, and glutamate 276. And this hydrophobic pocket accommodates the glycerol side chain of cenobamate. We evaluate the effects of mutations at residue 246 on cenobamate binding using the trimutation function in ICF-Pro. Substitution of alanine with valine or freenine served as positive control. As expected, substitution of alanine with valine or freenine at position 246 resulted in a large free energy change of 5.57 and 5.18 kilocal per mole respectively. On the other hand, the substitution of alanine 246 with serine resulted in a non-significant binding free energy change of negative 1.80 kilocal per mole. This suggests that the sequence variation of residue 246 in wild-type neuraminidase have a minimal effect on cenobamate binding. We also explored a resistant mechanism of the neuraminidase protein against a current inhibitor. As mentioned previously, during the 2009 swine flu pandemic, a histidine 274 tyrosine mutation was observed in the neuraminidase protein of the circulating strain at that time. Although residue 274 does not line the calendar pocket or directly interact with the bound ligand, the histidine 274 tyrosine mutation was shown to confer oseltamivir resistance in group 1 neuraminidase. Here, the superimposition of the wild-type neuraminidase and the histidine 274 tyrosine mutant neuraminidase is shown. The substitution of histidine with a bulkier tyrosine residue at residue 274 pushes glutamate 276 further into the oseltamivir binding site. As a result, the normally hydrophobic interaction between the pentoxy substituent of oseltamivir and the beta carbon of the methylene group of glutamate 276 is disrupted. The second protein that will be discussed in detail today is the ns2 protein, otherwise known as NEP. The ns2 protein is a homodimer that contains two functionally distinct domains. The N-terminus domain from residue 1 to 53 contains a nuclear export signal motif, which interacts with the cellular export protein CRM1. On the other hand, the C-terminal domain from residue 54 to 121 binds to the nuclear localization signal of M1. Here, the crystal structure of the C-terminal domain of ns2 is presented. The ns2 protein is a multifunctional adapter molecule. Two major functions of NEP have been categorized, including facilitating the nuclear export of viral ribonuclear proteins and regulating the transcription and replication of the influenza A viral genome. The figure on the right shows the hypothetical model of the nuclear export complex. The viral ribonuclear protein complex, which consists of the viral RNA, MP, and the polymerase subunits PA, PB1, and PA-P2, is bound to the C-terminus of M1. The N-terminal NL signal of M1 is in turn masked by the C-terminal domain of NEP to prevent nuclear re-import. The N-terminal domain of the NEP interacts with the export protein CRM1 in a RAN-GDP dependent manner, and this allows for the nuclear export of newly synthesized viral RNPs. However, the biomechanism behind both functions is not well understood. In our study, we have failed to identify any potential binding pockets of ns2 using ICM-Pro. Therefore, we are unable to continue our druggability and genetic analysis for this protein. Combining the data from my lab members, we compared the genetic conservation of all binding sites in the influenza A proteome. Here, we plot a graph of the percentage of residues lining the pocket with a percent identity of over 99% on the x-axis against the percentage of residues lining the pocket with a percent conservation of over 99% on the y-axis. Each dot represents a binding site in a given influenza A protein. Looking at the top right of the figure, the endonucleus and C-terminal domain of PA, the cap binding and C-terminal domain of PB2, and the catalytic pockets of NA display the highest percent identity in conservation across all influenza A viral strains. The endonucleus and C-terminal domain of PA, the cap binding and C-terminal domain of PB2, and the catalytic pocket of NA are ideal targets for development of a pan-influenza A inhibitor due to their high percent identity and conservation of residues lining their respective pockets. There are currently 2 clinical phase 3 drugs that target either the PA endonuclease domain or the BB2 cap binding domain, and these are baloxavir, marboxil, and pimodivir, respectively. As previously mentioned, the neuraminidase catalytic pocket is targeted by oseltamivir, zanamivir, peramivir, and lemivir. These neuraminidase inhibitors share a similar chemical scaffold with a 5 or 6 carbon ring and chemical substituents bending from the carbon ring. Future drug design for pan-influenza A neuraminidase inhibitors should explore changing the chemical substituents around the carbon ring of the neuraminidase inhibitor. There are currently no drugs that target the C-terminal domain of either PP2 or PA. Therefore, these 2 novel protein targets should also be explored in the development of a pan-influenza A inhibitor. To conclude, the influenza A virus poses a major health threat, and there is a need for the development of a pan-influenza A inhibitor. The catalytic pocket and hydrophobic cavity of the neuraminidase protein are both biologically relevant and are druggable targets. We also determined that the wild-type genetic variation at residue 246, either serine or aniline, lining the catalytic pocket, has a minimal effect on the binding of the neuraminidase inhibitor zanamivir. Resistant mechanisms may arise in residues that are not lining a binding pocket. The genetic conservation of these residues should also be considered in the development of a pan-influenza inhibitor. Finally, we identify the C-terminal and endonuclease domain of PA, the C-terminal and cap binding domain of PP2, and the catalytic pocket of NA to be ideal protein targets for the development of a pan-influenza inhibitor. Thank you for listening to my presentation.